Hi, my name is Alissa Schurr, and I am a third-year medical student and academic medicine scholar from NYIT College of Osteopathic Medicine. My research study is entitled Mutation of Polycystin in ADPKD is implicated in aberrant skeletal muscle phenotype in zebrafish. Autosomal dominant polycystic kidney disease, or ADPKD, is an inherited condition resulting from mutations in the PKD genes, which code for polycystin. The clinical hallmark of this disease is enlarged renal cysts. Zebrafish are widely used to model ADPKD, but interestingly, they also develop a dorsal axis curvature, or DAC, of the tail, the significance of which remains unknown. The aim of this study was to further elucidate the significance of DAC in PKD mutant and morphin fish through analysis of myotomes and birefringence patterns. Based on previous studies, we hypothesized that DAC in PKD morphin fish is due to a reduced rate of elongation of the dorsal myotome, that the PI3K signaling molecule has involvement as well, and that the polycystin molecule plays a developmental role in the overall phenotypic structure of the zebrafish. For this study, we injected zebrafish embryos at the 1 to 2 cell stage with polycystin or PC2 morpholino, which mimics the effects of a PKD mutation. We used various methods of imaging these embryos, including compound microscopy for morphometric analysis, immunofluorescence with confocal microscopy, and analysis of birefringence patterns with a polarizer. For any statistical analysis, we used students' t-test. First, we analyzed the myotomes of control and PC2 morpholino-injected or morphant fish and found that the dorsal myotomes of PC2 morphants had a significantly decreased rate of elongation compared with the control fish as early as 1 day post-fertilization, or DPF. The PC2 morphants also had shorter total trunk lengths and taller myotomes, meaning that they do not properly elongate their bodies during growth like the controls. In Figure 6, we used LY, which is a PI3K inhibitor, to treat embryos and analyze any effects of PI3K inhibition on growth of the ventral, center, and dorsal myotomes of the fish. For all segments, the LY-treated embryos were shorter than the controls and were delayed in their rate of growth. Within the LY-treated fish, the dorsal segments were shorter than the ventral and center segments. Collectively, this showed us that PI3K inhibition also prevents these fish from properly elongating during growth. Next, we further pursued any potential effects of PI3K inhibition on dorsal axis curvature. By using immunofluorescent staining and confocal microscopy of GFP transgenic fish, we were able to assess the myotomes and nuclei of control fish, PC2 morphants, and fish treated with LY. We found that the quantity of nuclei per muscle fiber did not differ between the ventral and dorsal segments of the PC2 morphant fish and the controls. The LY-treated fish had a significantly reduced quantity of nuclei in their dorsal segments. In Figure 8, we treated PKD mutant and morphant fish with LY to assess any potential interactions between polycystin and PI3K. The PC2 morphant fish treated with LY produced greater angles of dorsal axis curvature than those of both the morphant fish alone and of the control fish. However, the PC2 mutants, both with and without LY treatment, did not produce curvature that differed from the control fish. We were limited here by a lack of phenotypic penetrance in a mutant fish due to an issue with the fish breeding. Next, we analyzed birefringence patterns of the zebrafish. We had actually intended to use this modality to visualize the kidneys, but we noticed an impact of the PC2 knockdown on birefringence of the skeletal muscle tissue, and thus pursued this further. For control and PC2-MO fish, we imaged them at 1, 2, 3, and 6 DPF with a polarizer, and found that by 2 days post-fertilization, birefringence was present along the skeletal muscle flank. In the morphin fish, they had an interesting pattern of absent sections of birefringence associated with their axis curvature, which was not seen in the controls. In Figure 10, we imaged fish with a mutation in the PKD gene and noticed that the same pattern was present. This confirmed that this finding was not simply due to the morpholino injection and is associated with the knockdown of polycystin. On the right in Figure 11, we assess the temporal activity of polycystin during embryonic development. Here, embryos were injected with a PC2 photomorpholino, which allowed us to halt polycystin function at various time points by exposing the embryos to 360 nanometer light. We inhibited polycystin function at 3 different time points and found that the earlier polycystin was blocked, the greater the angle of dorsal axis curvature the fish would develop. This told us that polycystin is active early in embryonic development and may play a role in muscle maturation of the fish. To conclude, from this study we found that DAC in PKD2 morphin zebrafish is driven by a decreased rate of elongation of the dorsal myotome as opposed to decreased nuclear proliferation of the dorsal myotome segments. Also, the trunk lengths and heights of the PKD2 morphin fish suggest that the underlying mechanism of DAC is related to a defect in convergence and extension. The PI3K signaling pathway is likely involved with this phenotypic manifestation as well, but more investigation is warranted there. To our knowledge, this study is the first to suggest a direct effect of defective polycystin on aberrant skeletal muscle maturation. So, we propose that polycystin plays a novel developmental role spanning multiple tissues. Unfortunately, due to the COVID-19 pandemic, we were not able to go further in depth into analyzing potential interactions between polycystin and PI3K the way we were hoping, so this remains on our to-do list. Thank you for listening to my presentation.